So the title of our project is Precision Irrigation in Nursery Using Wireless Tensiometers. It's a project that I'm doing in collaboration with Professor Jean Caron and Professor Steve Pepin, both from Laval University. So before going into the result of the first year, I will describe the general context of the project. Growers are more and more aware of the importance of optimizing water use in nursery plant production. The motivation behind this priority are going from the increasing cost of the resource, limited water availability for some nurseries, and a general concern about the environmental issues. The ultimate goal is to reduce water use while maintaining an optimal growth of the plants. In other words, put the right amount of water at the right time. So we're going to go to the next slide. The first challenge to achieve this goal is how to decide when to irrigate. There are multiple options to answer this question. The one we have decided to investigate in this project is the use of wireless tensiometers, combined with the identification of the optimal irrigation threshold. I will come back to this technology in a few slides. The second challenge, as described in the next slide, is how to adjust the irrigation with a wide diversity of species. Growers have often hundreds of species to grow, and it's hard to take care of each species separately. It would also be expensive to put wireless tensiometer in all of them. For this reason, we are exploring in this project how to create the best species clustering based on their water needs. Let's go back to the tensiometers on the next slide. For those of you who would be less familiar with this instrument, here's a simplified figure that illustrates how the tensiometer measures the water available in the soil. Briefly, there's water in a column with a porous tip. When the soil dries, either because of evaporation or because the plants are taking the water is pulled out of the tube. It generates a vacuum at the top of the column that is measured by a gauge. The measure gives a precise indication of the water available to the plant. On the next slide, I show you the new generation of tensiometers by the company Ortho. By sending the data by cell phone signal, they allow a real-time monitoring of the soil moisture. The data are available on the web interface, and alarms can be set to notify the growers when the plants need water, either by text messages or by email. It is also possible to connect the system to automatic valves to control the irrigation. On the picture, you can see the box that collect and send the data. You can connect up to 8 sensors to each box, and on the picture on the right, you can see the sensor that would be inserted in the soil with the ceramic tip. At the bottom of the slide, you can see part of the web interface. That's okay. So in this slide, the one you see right now, in this experiment, we have decided to try 4 irrigation thresholds based on the metric potential. For metric potential, a positive value indicates pressure, while negative values indicate suction or tension. In a soil, the metric potential is normally negative. When the soil dries, the metric potential becomes more negative. It is important to point out that you can also express the value as a tension measure. In that case, the value becomes positive. The web interface of Ortho is using the positive value. But regardless, we have decided of 4 thresholds based on the substrate we use. A nursery mix from Fafard that is pretty standard in the industry. At -12, the water is almost not available, but it is before damages occur for most plant species. So that's why we decided to use that drier treatment. At -3, the soil still contains a lot of available water. So it was the 2 extremes that we chose. And the 2 other thresholds are distributed in between. So next slide, please. We were able to monitor the tension by using the web interface. When the threshold was reached and irrigation was done, and the tension go back to almost 0. The different colors represent the 4 irrigation threshold. And as you can see, we were trying to really get into that zone, but sometimes because we had to do irrigation just before, at night before leaving or early in the morning, sometimes we were going a little bit over or under, depending of the time of the day. As you can see on the figure, the tension can change quickly depending on the weather conditions. It was therefore important to monitor closely several times per day and go water every time we needed. So next slide, please. For the first year, we used the 4 thresholds, that I just told you about with 10 reference species. And those species were really, we really used them to really represent the most important species in nursery. At least we were trying to do so. So we did the experiment under a tunnel to control completely the irrigation. And you can see in this slide and the previous slide a setup of the experiment for that. For the 10 reference species, we have selected 5 perennials and the 5 shrubs that are popular and have different water needs. You can see the cultivar names of the species we have selected here. The experiment started with plugs, and we used 2-gallon pots for all the species. In our selection, we also tried to take species with different growing speed to have the best diversity possible. So in the next slide. In terms of data collection, we did measure the water used with water meters. We also recorded the grow parameters several times during the summer, and at the end of the season, we harvested the aerial part of the plants and used a drying room to measure the dry weight of each replicate. So it really allowed us to really have the maximum amount of data for each. And we also did look at the appearance of the plant, so we would be able to sell the plants during the season. So on the next slide, you can see some pictures of the setup. In the first picture, we see the water meters with the manual valves to control the irrigation. In the center, you can see the setup for one species with all the replicates. On the last pictures, we can see the micro sprinkler that was used to water the plants, and also the top of a tensiometer probe. So you can see it's a little bit outside of the soil, so you can really see it well, and there's a handle that you can remove it if you need to during the season. So we will now look at some of the results with the reference species. I decided to show you 3 species out of the 10 to illustrate the differences we saw between the treatments. The first thing I should say is that for most plants, all the treatments gave plants with a good appearance at the end of the season. There were no major damages to the leaves, for example, and it's not unexpected with the threshold that we decided to use. However, for most of the species, we saw differences in the biomass. That means a grower could decide in theory to go from one threshold to the other depending on their specific condition. In other words, if there's a shortage of water, they could go for the drier treatment, knowing that growth will be slowed down, but without major consequence on long-term appearance of the plant. Since growers often want to grow the plant as quickly as possible to send them to the market, we have based our analysis on best growth in combination with the lowest water use. In the first example, we will look at the data for the cedar. As you can see on the figure, the treatment with the wetter threshold gave the best growth. In fact, there is a 50% increase compared with the -12 treatment. The drier treatment could still be used to reduce water use, but there's a price to pay in terms of biomass. So we can see a picture of representative plants for the 4 treatments. And as you can see, they all look good, but the -3 is better looking than the 3 others. So in the next slide, we have the result for the Astilled. In this case, we saw some damages to the leaves for the -9 and -12 treatment that would reduce their value on the market. So they are not good option in this case. It was the plant with the most sensitive, that was the most sensitive to water deficiency in our assay. The 2 treatments that also have lower biomass yield compared with the other treatment. Between the -3 and the -6 treatment, there are no major differences in the biomass yield, and the plants have no damages at all. Based on these results, we have selected the -6 treatment as the best one, the optimal one. If we look at another example in the next slide, for the spindle, the best treatment was -9 with a good biomass yield and a lower water use compared to the -3 and -6 treatment. Compared with the -3 threshold, it allows a reduction of 5 liters per plant during the season. And it doesn't seem a lot, but if you look at in terms of percentage, it's about 30% less water use. So it can represent a lot of water if you have like a big surface with a lot of plants. With those 3 examples, you can see how the optimal threshold can vary from one species to another. One of the most important elements to point out is the great difference in water use between the species for the same threshold. This illustrates how important clustering could be. And for example, the Astilled at -3 used 80 liters during the season, while the Asta used only 16 liters for the same threshold. This is mostly associated with speed of growth and the transpiration level of each species. in the next slide, please. If we look at the objective of the first year, we were able to identify the optimal threshold for 10 reference species. You can see the value on the table. I should point out that we are still debating the best threshold for some of the species based on all the factors. Sometimes the results were really close, so we really want to analyze everything to decide what is the best one. We will finish analyzing all the results in the next few weeks. So the results from the first year will be used to do the second part of the project that I will now present you. There are two main goals for the second part of the project. The first one is to identify the best clustering for a wide range of species. We will associate them with the 10 reference species that we did this year and that cover a wide range of water use. For example, based on the result of the first year, a group associated with the acid would receive around 80 liters of water during the season. For the group where the tensiometer is in the spindle, they would only receive around 12 liters, so there's a big difference. At this point, we do not anticipate any major challenge that would prevent us to perform the next year experiments. Here you can see an illustration of the clustering experiment. So it's only an illustration. It would not be exactly like that in the setup, but it's just to show you. So each year we will test 29 species and we will combine them with the 10 reference species, with 3 replicates of the treatment for each. At the end, we will be able to tell, for example, if the species 21 should be grouped with the acyl or with the spindle, for example, as illustrated here. So we're going to go to the next slide. The second goal is to compare different irrigation strategy in a commercial nursery. We will use one of the reference species in 2016 and a cluster of 2 species in 2017. In the next slide, I'll present the 4 treatments that we will use for that step. So if we go to the next slide. Yes, thank you. So the first one is conventional irrigation by the grower. So it's simply what they do normally. The second one is with a tensiometer and an automatic valve controlled by the software. If we choose the acyl, for example, the irrigation would be done at -6. The third one is with a capillary mat controlled by the grower. Capillary mats are known to reduce water use by decreasing water leaching, and they provide a constant water supply to the plant. So it could be a good option here. The last treatment will be a combination of capillary mat and tensiometer to try to optimize and try to see if it would increase the water saving. The setup should allow seeing what treatment use the less amount of water and what is the impact also on the growth of the plant, so the speed and also the biomass at the end of the season. So we're going to go to the next slide. Thank you. Now for a look at what will be the result for the industry. At the end of the project, we should be able to provide a clear guideline to the growers that includes the optimal threshold for the reference species and the best clustering for a wide range of species. The grower will also be able to compare 4 different irrigation strategies for water use and the impact of those treatments for the growth of the plants. So next slide. For the knowledge and technology transfer, our strategy during the project is to present the preliminary results to the growers by conference. For example, I did present the project to the Quebec Association of Nursery Growers last February. And we also do meeting with the partners of the project on a regular basis to share the results. At the end of the project, we will or when we will have enough data, so it's going to depend how it's going to go with the second and third year, but we will share broadly the results with the Canadian industry. We will use publication and also conference to do so. And we have not decided what journal we're going to use or what magazine, so we're going to decide that depending on the result and if they accept to publish our result, of course. So if we go to the next slide. I would just like to acknowledge all the partners for this project and the entire team at Laval University, and thank you for your attention.
Great. Thanks very much, Charles. Any questions for Charles?
It's Jamie here. Were the fertilizer rates, those ones used by industry, were that part of your trial?
Yeah, so we did use the fertilizer because the second and third year we're going to go to Quebec Multiplan. that is the grower, the commercial nursery that we're going to go, and so we use exactly what they use in their treatment, and I don't have the exact number here. I'm sorry, I should have put it beside me, but so we use exactly what they do use normally.
Okay.
And it's a slow release fertilizer.
So the only factor was the water, not the fertilization rate.
Exactly.
Okay.
Charles?
It's you being here, Charles. Maybe I will talk to you after this. Maybe we can collaborate
That would be
to do
great.
some work. Yeah, because we are doing fertilizer rate, and we want to look at irrigation level as well. So I'm going to contact you. This is great.
Definitely.
Their research is just marvelous.
Thank you.
Yeah.
I have a quick question. It's Mike here. You were mentioning the work with Thuya, with the eastern white cedar. That's a species. That's the species that I know and love, sort of. But in our work, we found that basically you could give it as much water as you could possibly afford, and it responded positively. But one of the questions that we're starting to consider is what is the conditioning effect of for example, a 3 kilopascal or whatever that unit was, but what is the conditioning effect of that if you have a nursery crop conditioned to very luxurious water application rates, and then you stick it out on one of Darby's roadsides? The inevitable consequence, as we have seen historically, is that they don't survive.
That's a really good point, and that's something that we are thinking about. And I'm not sure I could answer all the question here, but I'm pretty sure that if you have a drier treatment, I'm pretty sure that the plants will look better after either in a garden center or at people places, because they don't have ideal conditions there. So yeah, that's a really interesting point, and I don't know if we should put emphasis on the best growth possible and then start to do kind of an acclimatation to help them to do the transition. I don't have the answer for that, and maybe you did some work on that. I don't know if you can help me on that side, but that's
we are definitely making progress on that particular issue using the automated wireless stem psychrometers for measuring total water potential in the plants. It would be very interesting to correlate the plant's response to irrigation management, which is what we're doing, to the soil water status, and correlate those, because that's the next step in our research as well. So it's quite interesting.
Yeah, definitely.